Welcome, everyone. I'm Jen Farmer from the Friedrich's Ataxia Research Alliance, and so glad you can join us today. I'd like to begin by just quickly thanking everyone in the FA community for your engagement and support during FA Awareness Month. There was just a lot of different activities to participate in this month, from the flash talks to webinars to social media campaigns, as well as activities in people's local community and fundraising events. And it was really a great month. And just want to thank everyone for your ongoing engagement and support. So welcome to our webinar today with Solid Biosciences. I am joined by Seth Rotberg and Matt Harmelink from Solid, and they are going to give you an update on the SGT-212 program in Friedreich's ataxia and the ongoing clinical study, the FALCON study. We'll get updates from Seth and Matt, and then we'll open it for Q&A and just ask that folks put their questions in the Q&A box, and we'll be moderating those and do a moderated discussion after the presentation. So with that, Seth, I'm going to turn it over to you.
Awesome. Thanks so much, Jen, and thank you everyone for joining us. For those that are based in the US, I hope you all had a good Memorial Day weekend and really just excited to be here. As Jen mentioned, we're providing an update about our SGT-212 program for those with Friedreich's ataxia. It's the FALCON study, which is a phase 1b study. And, we really just appreciate this also because it is FA Awareness Month. And we're just excited to provide this update. So, Matt, if you go to the next slide. So, I do want to just kind of kick us off by briefly introducing myself. As some of you may know, I'm the Senior Manager of Patient Advocacy at Solid. I got involved in this space about 18 years ago after learning about my mom's diagnosis with a rare neurological disease called Huntington's disease. And seeing what she went through for 17 years is what really fueled my passion to get more involved, really to help the community and really help infuse that patient perspective into research. And so during this time, I really learned about the importance of working in partnership with the community, with patient advocacy organizations to really make sure we're driving research in the right direction. And so we do appreciate partnerships such as the one we have with FARA, to really help us work alongside and making sure we can provide updates to the community, such as the one today. And so in this webinar, you'll not only hear from myself, but you also hear from my colleague Matt Harmelink, who's our Senior Medical Director, Therapeutic Area Head of Neuromuscular. And Matt will be the one really diving deeper into the study. And then, as Jen mentioned, we'll leave some time at the end for some Q&A. If you go to the next slide, just kind of quickly, we'll share that, Solid Biosciences, we're a publicly traded company, so we do need to provide a forward-looking statement here. Next slide, Matt. And so just a brief overview of us. So we are a precision genetic medicine company. We're focused on gene therapy in the neuromuscular and cardiac space, specifically in rare disease. And the company was founded in 2013 after two parents learned about their son's diagnosis with Duchenne muscular dystrophy. And since then, we originally, our focus was in Duchenne, but we've expanded to other rare diseases such as FA and a rare cardiac disease known as CPVT, and then a few other kind of rare cardiac conditions. We do, as mentioned earlier, work closely with patient advocacy organizations from around the world to really make sure we're including the patient perspective in our everyday work. Solid is based in the Boston area, and our leading clinical trial is for Duchenne, which has a phase 1/2 study and a phase 3 study, followed by our phase 1b study for FA, as well as a phase 1b study for CPVT. And so, with that being said, I'm excited to pass it on over to Matt, who's going to dive deeper into our study and provide the latest update of where we're currently at.
Matt, you're on mute.
Thank you. Sorry, I obviously am not the tech person here. But no, thank you so much for having us. We really do appreciate the ability to sort of talk about the program and the study. I'm going to start us off with sort of what is the overview of our investigational gene therapy, SGT-212. SGT-212 is an investigational gene therapy designed to address the neurologic, systemic, and cardiac manifestations of Friedreich's ataxia. For those of you who maybe have not seen, we did do two previous webinars where we actually talked about the construct itself, some of the science behind gene therapy, and I'm not going to cover that again today. But if you're interested, that is actually up on the FARA webinar for your review as well. For us, SGT-212 was designed to deliver this full-length frataxin gene into certain cells in the body. And it's being done by being administered by two routes. There's an intradentate nuclear infusion into the brain and an IV infusion in the arm. The intradentate nuclear infusion allows the delivery of the therapy directly into the part of the brain, the dentate nucleus, that is most affected by Friedreich's ataxia. And then the IV administration can address the largest cause of mortality in FA, the cardiomyopathy, and also offers a potential to treat other organ systems that may be affected by Friedreich's ataxia. Participants in our study are followed for around 5 years after dosing. And as I go through, I will walk through a little bit about the overall timelines of what the study looks like, some of the inclusion criteria, and we'll talk more about this ID and infusion and try to demystify it as we discuss a bit. As we start off with any type of trial, we know that SGT-212 has been evaluated for the treatment of Friedreich's ataxia patients by preclinical studies. Preclinical studies are important because before we get to the clinic, we want to evaluate in animal models, in this case, mice and non-human primates, both the route of administration, safety, and potential efficacy. The preclinical studies that we did in mice and these non-human primates or types of monkeys and primates confirm that administration of SGT-212 can support that frataxin expression in target tissues, increase mitochondrial function, improve cardiac and neurologic function, and increase survival in the animal models as well. And that's important then as we start to move forward into our human studies and sort of what we're seeing currently in our FALCON study. As you'll see on the right, a very upper part of the screen, that is the FALCON study that I'll be talking about. And in our human study, the study criteria at a high level that we're talking about is we are currently enrolling patients who are either male or female, age 18 to 40 years of age at screening. The study has started with non-ambulatory participants with the plans to move into ambulatory participants as we go further along. And participants coming into the study cannot have received any gene therapy at any time. They also have to be negative for AAV antibodies. These are antibodies that potentially could limit either the efficacy or potentially could affect the safety profile as we evaluate the potential medication and treatment. And so those are part of the screening process I'll talk about in a bit. As you can imagine, patients would have to be diagnosed with Friedreich's ataxia and have symptoms start at 25 years of age or younger. There are other objective functional and cardiac criteria that are involved. Some of these are listed on clinicaltrials.gov, and for others, study-specific criteria, you can certainly chat with a study team if you're interested about how this study may relate to your specific case and symptoms. What's important also is that if you're taking Skyclarys or omaveloxolone, you then need to be on a stable dose for at least 12 weeks coming into the study and continue taking that dose throughout the study. If you're were on it but stopped it, you will need to at least been off for 12 weeks before coming to the study since the last dose. Also coming into the study, participants can't be on other investigational or approved disease-modifying therapies. And again, if you have any questions about very specific criteria, I'd encourage you to look at the clinicaltrials.gov or also going into chatting with one of the study teams that I'll mention later on about specific criteria, as well as your neurologist about whether this study may be right for you. So as we think about the participant journey coming into this study, as I mentioned before, it's a 5-year study, but there are we can break it down sort of into different phases. We start off with what we see as this pre-screening. So a participant coming into the study, we want to make sure they would meet the criteria, as I mentioned, the objective criteria amongst others, to come into the study. And that takes up to 45 days before dosing and includes that AAV antibody testing that it was mentioned. As participants then come in, they go through this with medication reviews, of physical exams, lab testing, these initial assessments before they get to day one, which is our day of dosing. That would be where the participant would have the ID and surgical infusion, followed by that IV infusion. And then they're closely monitored for that period with safety in a short hospital stay. That's really just to make sure at post-dosing that they're doing well, that we can closely monitor any participants in the study. From there, they would move into this frequent outpatient visits, where it's again a lot of physical exams, laboratory testing, and really close monitoring to meet that objective primary goal of safety and tolerability of SGT-212. And that's in this period here of day 6 to 60, but then also moving into, as you saw, sort of wiggle a bit into the day 90 assessments, our primary safety outcome at month 12, and then also month 12 other assessments. And that's important as we move through to understand the longer term effects as we see them of SGT-212 in humans. As we get beyond that initial 18 months down to year 2, you'll see down here we have then these periodic outpatient follow-ups that occur for years 2 to year 5. Every 6 months, it's visits with a physical exam, lab testing, functional assessments, side effect review, and medications, but also once per year, also heart and brain imaging as well, and followed through to the end of the study to understand the longer term effects of SGT-212 in humans. Now, I had alluded to a bit that I wanted to talk more about the surgical approach that we're doing, this intradentate nuclear infusion. The approach that is a neurosurgical infusion, and as you'll see on the left panel, and from the title, this is a robotically assisted brain infusion to help with neurosurgeon administration. I will admit I'm not a neurosurgeon, so specific questions I will avoid, and those are all things that would a participant want to talk with the site and study neurosurgeon before they move forward with the study. But in general, as participants come into the study, they have, prior to surgery, imaging of their brain MRIs, for example. That can be mapped over in the OR, other imaging, to actually show, and I'll mention this in a few panels in the slides, the dentate nucleus of a participant. From there, the robots can actually help direct a trajectory of where the catheter that would deliver the SGT-212 into the dentate nucleus should go. And you can actually see that on the left panel, where you see the array, as where it's sort of pointing down to that back part of the brain. Once that is in place, the surgeon can actually then make small incisions and actually put a small bolt piece that will actually hold the catheter in place into the back of the brain. And there's a dentate nucleus on both sides of everybody's brain in the back. And so there's two placements that they do with two catheters. The catheters are then advanced into the brain tissue. And you can see that here of one catheter is in place in the middle panel on one side, and one is being placed on the left side going in. At that point, the SGT-212 can be infused into a participant in the study. And throughout the infusion, there are periodic MRIs as well that actually can then look and evaluate at where is the actual infusion of the SGT-212 in the brain. This is why we call this a precision MRI guided infusion. And we do this is being done to treat the dentate nucleus, that part of the cerebellum that's a key focus because it's heavily affected in Friedreich's ataxia. The robotic assisted placement and the stereotactic approach is important because it allows the precision piece of this infusion. After that has been done and the infusions are complete, participants would then get the IV infusion of SG212. This is a systemic infusion with the approach designed to enable delivery of the therapy to the heart, a key organ involved with the cardiomyopathy, that leading cause of mortality in FA. And I apologize for the worst image here. I think most people are very familiar with IV access and infusions at this point. So where does that leave us then? So we look at our study overview. So this is going back to the high view of the study. This is the FALCON study. It's a phase 1b, meaning that it is a study of participants who have Friedreich's ataxia. Open label, meaning that everyone would get the medication, multi-center trial focused on evaluating the safety and tolerability of SGT-212 in patients with Friedreich's ataxia. Currently, we have 3 study sites. Ohio State University is actively recruiting, and UCLA, CHOP UPenn are very close, but not yet recruiting at this point as we go forward. Again, updates to this will be continuously put on clinicaltrials.gov. So if you have questions as we go forward after the webinar, please feel free to look at that site for both these sites as well as any others that may become involved in the future. Now, I wanted to bring this back a little bit to our first participant in the study. And as we look at this first participant dose in the FALCON study, this is actually his intra-procedural MRI that confirmed the precise delivery of SG212 in the dentate nucleus. On the leftmost panel, this is a baseline MRI, so pre-dosing, pre-surgery. For those who don't read MRIs all the time, MRIs have light to dark scale in terms of how we look at things for most of the sequences. the front or the top of our head where our nose is, you can actually see labeled front for ease. And the dentate nuclei of this participant is actually circled in blue for easy visualization. That image was then can be merged, as I mentioned, in the OR pre-dosing with SG212, which is our middle panel. So again, you can see the front here. It will look slightly different because the sequence of images are sometimes a little shifted in terms of the one that we're using and showing here. So if you don't see as much of a prominent nose, that's okay. You will also see again where it's been outlined the dentate nucleus to give that ability to see it on the image, as well as the fixation devices, what I mentioned, where they put the catheters through that go into the back part of the brain here, and there are not catheters present. Then as we go to post-dosing, over on the rightmost panel, again, you can see where the fixation devices still are coming through in the imaging. But what's important to see is SG2212, the actual drug product, you can actually see here very bright. It's bright and it's beneficial that way, as this is what we would see on the multiple MRIs that are being done through the course of the surgical procedure, that the surgeon is able to follow, track, and then give that precise delivery of the drug as we go forward. As of February 2026, this is the initial safety observations for our first participant that was dosed. Cohort 1, or a non-ambulatory cohort, we had one participant that had been dosed as of this cut date. We have announced that we've had our second participant dosed currently. For this participant, though, at this time, he had no serious adverse events and no treatment-related adverse events. As of the day 45 visit, there were no treatment-related adverse events, with only one surgical procedure-related adverse event of headache that was reported. And I'm seeing a lot of questions popping up. So I'm going to, as we come to the end of sort of the initial talk, wanted to sort of take this moment to pause again to thank the community for information on the study, but also open it up to Jen for other questions that may be coming up that I can address as we go through.
Matt and Seth, thank you so much for the update and the overview. We did put in the chat the links for the prior webinars so that if folks want to go back and learn more about SG212, there's a lot more information about the gene therapy in those prior webinars. There are a few questions coming in from folks related to the inclusion-exclusion criteria. And so one question was related to their genetic status related to FA. So is the study open to people who are compound heterozygous, who might have a GAA repeat expansion on one allele, and then some other type of mutation, whether it be a point mutation or a deletion or missense mutation on the other allele?
So the study would be open to, and this is something you'd want to talk with your study team, to more than just people who have repeats on both alleles. Is something that more details can certainly be given if someone reaches out to the study sites about that. But I would encourage people to think beyond the repeat expansions only.
Okay. Good news. The other questions related to criteria, I see some questions coming in related to their plans for sites outside the US. And I know I've gotten questions in the last few weeks from people ex-US asking if they can participate in the study, if they're willing to travel to the US, if they could stay here for the time that's required for the procedure, the monitoring, and being able to attend subsequent visits.
Yeah. I will defer to the sites that are running the study about the specifics in chatting with a potential participant. From a study level, it is something we would allow, but we want to make sure that it's done safely, and each site has their own criteria. So if you're interested, I'd encourage you to reach out to the site to chat more about that. They can share more about that piece, as well as the status that they are in recruiting, to make sure we want to be very transparent to anybody interested in terms of timeliness of getting into the study, but also, making sure it's safe for them.
Thanks. And then I think the last question I saw on kind of the inclusion or the criteria is around people who might have late onset FA. The study criteria currently is targeted more to people who have typical onset FA with symptoms starting before 25 years of age. And
Mhm.
so folks are wondering if you'll eventually be able to open this to folks who might have later onset FA.
Those are great questions, just as the pediatric question, which we've gotten before about going into children as well. Those are all things that we want to be able to consider as we thoughtfully go through the study. This is a phase 1b study, so we're being very thoughtful in our approach as we gather data and are engaging with the sites, the participants, and the experts in the community to make sure that we're doing what's best as we look at the next step.
So maybe I'll move on to questions related to the study and the procedure itself.
Mhm.
There are a few folks asking some follow-up questions to kind of your explanation of the intradentate injections. One participant is asking if the participant is awake during the IDN infusion. And let's see, I'll find the other question real quick here. But the first one was, are people awake?
So in general, with these procedures, patients are not awake during any type of robotic infusions. So no.
And how long does the procedure take? What should people anticipate?
So this is something we've not, I'll be quite honest, we've not disclosed exact procedure length. And as we've looked at the first participants dose, it's something that we're continuing to follow and trend. It's something I'd want them to talk with their surgeon about because some of that is also very surgeon and participant specific in terms of how long they anticipate that to take. Just like with any procedure, there is a technical component and a person component of how do they best get approached by the surgeon for this. So I don't want to give any leading information for that.
Let's see. There, there was a question I saw quickly around how many participants. You had mentioned that now 2 participants have been dosed.
Mhm.
How many do you intend to dose? I know going back to the prior webinars, you presented to the community the initial study design of having kind of two cohorts, 3 participants
Mhm.
in each cohort. Is that still the same?
Currently, that is our study design. We have listed 10 participants total, 3 participants in cohort one, 3 participants in cohort two. Obviously, the math would be that there's a potential other participants as well in an optional cohort. That'll be something that as every participant is dosed and we gather data, we want to be engaged again with that feedback loop of with the experts, the participants, and determining how we look at the study overall. So while that is where it's at right now, I would encourage people, if they're interested, to stay engaged, because that may change based off the data, like with many studies.
Um, let's see. Oh, I just saw a few more questions around inclusion criteria. Can you share a little more about the criteria related to like cardiac status in FA?
Um, I can. So as we're looking at our participants, I don't believe we've said ex all the criteria. Some of them are on clinicaltrials.gov. We do want to have we want to make sure it's safe first and foremost for participants as a new drug. We also want to make sure that we're able to gather the information that we're looking for. So that's part of the cardiac MRIs that would be done as part of the screening process. We get data back, and then that site would be able to talk through with the participant about the whether they meet the screening criteria. If someone is interested in understanding if potentially beforehand, that is something that some participants have worked with the sites as well to ask those questions before of, do you think I'm a good candidate for that? And that is a great question to ask, or to engage with their cardiologist and neurologist about as well, to understand some of those potentiality. I understand as participants are looking at trials, you don't want to go through the whole screening process if you know you won't qualify, right? And so those are wonderful questions to ask that way. Um, that I think because of some of those nuances of those objective criteria, they can help ensure that they might be a good candidate before they come in, as I think they'll to go. If I can go round one back, and just because for those people who are very interested in the surgical procedure,
Yeah.
some are on the bottom of our, actually the citation here is actually the group that helped design the technique. It's a publication that came out. So if someone with FA is interested and wants to pull in their neurologist to read it, it is a public publication on the exact type of technique that people can can read about. It's a scientific paper. I will warn you with that. That's very technical from the neurosurgeons, but it may be interesting for those interested who want to have a conversation with their care team a little bit further.
Um, Kind of to follow up on those procedure questions, there are
Mhm.
some general questions that maybe you can give a little bit of context to in terms of like what what's the anticipated recovery? What are some of the risks associated with this kind of procedure?
Yeah. Again, I'm not as a non-neurosurgeon. I'm a neurologist by background and training, so a little different approach to it. Um, this is something that while we are watching very closely, right? This is a phase 1b study, and I apologize if I say that over and over again. We want to watch participants closely because it is a first-in-human type study. On that same note, as we go forward with the procedure, it's something that, as you saw from the first participant's experience, what I can mention here is adverse events are reported from participants when they have something that they feel is not right. And this is not just, as you see here, it's called treatment related or treatment emergent. There's different terms for different types. In a study, we encourage everything to be reported because we want to know the broad scope of it, of things, even if someone doesn't feel they're related. After you look at the study and all the data, you can incorporate that. For this first participant, what they reported was no serious adverse events, and there was this one surgical procedure, a adverse event of a headache that was reported. So hopefully that gets a little more context of that's what we gathered in terms of sort of that participant's post-dosing experience. We do mandate, as you saw before, that patients stay in the hospital just for that close monitoring period. That's all participants, no matter how they respond to the medication.
Um, I would also just encourage folks who are interested in the study. There's also, before you sign up for any kind of research study, there's a process that you go through called informed consent with the study team. And this is a document that, in a lot of detail, goes through everything that's known, everything that's planned about the study, including potential risks. And
Mhm.
there's, risks that are reviewed as part of the informed consent, both related to the procedure as well as the treatment, as well as to other tests and evaluations you might have, like those MRIs. So this is a document that is really valuable to you in learning more about the study, especially the procedures and the potential risks. And so would encourage anyone who's interested, you can even ask the study team to kind of review that with you as you're going through, the outreach. If you're not sure you want to learn more, they can help you with some of the key informed consent aspects.
Cool.
Let's see. So there's also a bunch of questions kind of on the flip side of risk about potential benefit, right? So people are wondering if we know anything yet about potential benefit, either from the first few participants or I don't know if there's anything from preclinical studies, Matt, that might, guide what people can expect at this
Mhm.
point.
Yeah, and I'll maybe start with the preclinical as I find that slide. Um, So in the preclinical studies, we were able, again, these are mice and non-human primates, not in humans. But we were able to see that there was frataxin expression, as well as increased mitochondrial function. We know the mitochondria is an affected part of the cell in Friedreich's ataxia. In addition, there were improvements in the cardiac and neurologic functioning based off these different animal scores. That if you're interested, there's actually down here an entire presentation on this. I will not go. I don't want to go through for everybody and bore you with animal and mouse models for those who are not interested. But there's data from those models on different neurologic scores, cardiac scores, and then also because we're looking at the cardiomyopathy and the mortality in animal models that increase survival. So from that data, we're able to move into the clinical space. We have not talked publicly about any efficacy data. It is very early, as you saw from the dates. And our public announcement of our first participant dose in January, it is very early still to be making those evaluations. Those are things that we're going to be continuously following as part of the study. And as once we feel we have more data and are able to publicly talk about that, we absolutely do want to come to the community as well, to have that discussion. But at this point, it's a very early stage in the study, I think, to talk about that.
Yeah, and just generally, phase one studies, which is what this is, a phase one study, are primarily designed to help us evaluate safety and dose. Efficacy is kind of in the secondary list of outcomes. And, it is, we'd like to have, a at least several people enrolled in the study and having completed a good amount of time in the study before we are really able to evaluate efficacy. So all
Mhm.
good questions, all things that I think we all hope to learn over the upcoming months and year. Um,
Absolutely.
let's see. Um, back to question about study design, and are you evaluating multiple doses, as we were just talking about kind of phase one, where we look at different doses? This question is about, will there be an evaluation of different doses throughout the study?
So that's a great question too. As Jen already alluded to, because it's a phase 1b study, that is a potential. I sort of alluded to there's an optional dose finding cohort. However, those are things we'll have to determine based off the data as we get it back. And so I leave that as a yes and question as we closely follow the participants along.
Boy, a lot of questions now. Let's see. Um, let's see. I also had a question that I saw from uh, Ron Bartek about the AAV antibodies. And I know when we previously have had webinars on gene therapy programs, this question comes up a lot. It's one of the inclusion-exclusion criteria. Um, is there, I guess maybe there are a few questions that frequently come up related to this is there an acceptable level of antibodies where people can still potentially be dosed? And
Mhm.
then another question that people typically have is, what's the frequency of folks having these antibodies? What's the likelihood that somebody will test positive for having antibodies?
Again, both great questions. There is a threshold cutoff for being positive versus negative to antibodies. Those are different based off the assay in the labs that people use if they're looking between studies or things like that. So it's something that is study specific oftentimes in terms of that piece, but it doesn't mean you can have none whatsoever. So I think so as people are thinking about this in terms of coming in, this is something to think about. But just also one antibody for one study is not the same as another. In terms of if you're looking between gene therapies, that's something to discuss with someone who knows your assays and knows you will have that discussion. In terms of the incidence in the population, again, we're just starting out with our study, so we haven't dosed enough patients to mention that. We know in the literature in general, and there's quite a few publications that have been published on antibody positivity. It can actually vary based off where you live. Some areas are more prevalent with antibodies than others, both rural to urban as well as just geography in the world. So we will have to see in terms of what that incidence looks like overall. As we look at other studies and publications, it can range though anywhere between 20 to 40, 50, somewhere in that type of percentages typically, again, because of these other factors. As we know more, that's one thing I'm certain we're going to be interested in knowing too, of what does that antibody positivity look like once we have enough participants who've gone through screening.
Um, Here's a good question, and I think it's an important question to the context of gene therapy trials in general. So this person is asking, why are we saying this study takes 5 years if the results, say, after 12 months are promising and, possibility to expand the study to more of the population like pediatrics or outside the US?
Mm,
Do we have to wait the full 5 years? Obviously, we wouldn't want to do that.
mm. Again, great questions. The 5-year period is really looking at that long-term perspective. With the gene therapy, the current feeling is you can only get one, which is why it's an exclusion criteria to come into our study having already received a gene therapy. And that's for a couple different scientific reasons. Because of that, we want to evaluate the long-term safety and potential if there's efficacy going down the road for that long period of time. As we start to gather more data, though, we don't have to wait for everybody to have gone through 5 years before we start to make the understanding of what do we think the effect might be. And then that brings it back to that informed consent process that was mentioned before of as someone who might be coming into a study in pediatrics or another group would want to understand how much data do you have so far? And was that the right study for me based off that data? I was a practicing clinician before my life at Salad, and some families and some people would say I want more data, whereas others are much more they feel they understand and would want to move forward or not based off less. And so that's all part of that informed consent process where data would come out and we would in a study would potentially move forward based off what that data looks like. As you can imagine, um, in both safety and in efficacy as we hear more about it.
Um, um, sorry, just quick going through. Miriam, if you see questions, my colleague Miriam is helping me monitor the Q&A. And so if there are some that you've seen I haven't gotten to yet, please help me out here because it's been a good activity in the Q&A box. Um, Matt, there is a question here kind of related to the interest in the study.
Mhm.
And I know this is something that would be helpful in terms of managing, expectations for folks. If you've already dosed two, are there a lot of spots still open? What should people expect if they contact one of the sites, given that this is an early phase one study with a
Mhm.
small projected enrollment?
Mhm. Yep. I think what I would encourage people if they're interested to do is if they are interested is to reach out to one of the sites or talk with their neurologist about reaching out to a site, such that we know if they're interested. That's always helpful from a site perspective, to know that. I do want to say we are being very thoughtful with the program, and we want to make sure that we are either A, not overpromising, but also moving very thoughtfully forward. So the site would reach out when they feel there's an appropriate slot available. I will say, and the FA community has been phenomenally supportive in terms of the amount of outreach that has occurred. And so if the site's a little slow to respond, I will apologize with them on they're doing their best in terms of engaging everybody they can. But we do know we've heard thankfully from Jen and Seth if there's participants who haven't heard something, and please feel free to reach back out again. Seth, I don't know if you want to add anything to that point.
Yeah, I'm happy to add. So, if there are any also additional questions, feel free to also you can reach out to we have a general email inbox. Excuse me. It's community@solidbio.com. And I'm happy to put that in the chat as well, unless Miriam beats me to it. But that's another opportunity where you can reach out to us and we can make sure to kind of support and make sure you get, the answers you need or any additional support and make sure you get connected to the right point of contact at each study site.
Thanks, Seth. Um, Maybe just one or two more questions here that I see. So this question is related to kind of the study design. And this is, since this is an open label study, are patients kind of serving as their own control, or is there a plan to compare folks in this study to an external control group like a natural history control?
That's a great clinical development question.
That's a Ron Bartek question. So
Thank you. So we've not publicly, very transparent, maybe not being transparent. We've not publicly disclosed how we're doing the comparisons as of yet, and those external comparisons. As a phase 1b study, we are very much focused on that safety and tolerability of the drug. And as we move forward, those are things that we are going to be engaging in more. And when we get to that point, sort of with the same with efficacy, that we can have that discussion and have a robust discussion, we're looking forward to having it. We're just not there yet early in the study to have those type of discussions.
I think we got to most of the questions, and I think that we all take away from a lot of the questions here the interest in the study, folks wanting to see things expand right to children with FA, as well as people in other geographic regions who've joined us today are expressing interest in participating in these kinds of studies. And, Matt, maybe we can just reiterate kind of what folks can expect in terms of upcoming communications, more information, and where they can get that.
Yeah. So as we go forward with the study, certainly we will be as transparent and giving out as much data as we can as it comes forward. We're anticipating an update in the second half of this year as we have some more participants who might be dosed and longer term safety data as we've talked about. That would probably be the next time. We're looking forward to engaging the community at other FARA events. For those who've attended those, they've probably seen me on stage a little bit, as well as like the CHOP FARA event, and then eventually sort of some of the scientific updates in the latter half of the year. Um, I hope this was helpful for the community. If it is, we're happy also to come back as we can have those discussions. I will say also thank you for the questions. They do inform for us as well where the community is interested in both hearing more, but also just understanding the thoughts and feelings that people have so we can really be engaged with what is the need and how do we help meet that.
Well, Seth and Matt, thank you both so much for joining us today and giving us an update on SG212 and the FALCON study. And we'll look forward to following up with you in a few months to learn more about how things are progressing.
Sounds great. Thank you, everyone.
Awesome. Thank you.